Hello, and thank you for joining us again at MIDD Plus 2023. In this session, Lisl Shoda, our Vice President of Immunology at our DILIsym Services Division, will present to you an introduction to our our COMPLEMENTsym software platform. As a reminder, please ask your questions in the Q&A tab on your dashboard at any time. If you need technical assistance, please use the chat tab just to the left of Q&A. Before I pass this off to the speaker, just a reminder that there is a poll question that we'd love for you to participate in. Thank you very much. Take it away, Lisl.
Great, thanks, Andy. And thank you for the opportunity to share some of our work on complement. I just have a standard disclaimer that we're not providing medical advice. And with that, I'm going to jump into what we're trying to do today. So with a pharma partner, Simulations Plus is developing a quantitative systems pharmacology model complement. The idea is for it to be applicable to multiple disease indications, and this includes diseases where complement dysfunction is sort of the primary driver of some of that disease pathophysiology, as well as diseases that have sort of a more complex nature, but where complement is thought to be a participant. And the way in which we're doing this is to create a base model that leverages an extensive body of publicly available data in healthy volunteers, but also in patients, and be able to customize that to particular diseases and particular drugs of interest. So in this talk, I'm going to spend the first part talking about why we're so interested in complement and why we think this is a great opportunity. And in the second part, I'll talk about what we've done to date and where we're headed for the future. So let me start with the basics. What is complement? Complement is part of the immune system. It really sits sort of in the innate immune response. And so in this graphic here at the bottom, we're looking at sort of the scenario where you have exposure to an infection. The first bit of the immune response will be the innate immune response, which operates on the minutes to hours type time frame and provides a rapid but sort of non-specific response. And in the second part of the immune response, you have the adaptive immune response, where you have a very targeted and pathogen-specific response. And it takes days to weeks in order to get that army mobilized and then get it out there and working. Complement is shown here on the bottom left, and it actually represents multiple proteins and regulatory factors. The other thing I'll just comment on is that the immune response in general is sort of easily depicted in all these different buckets, but it's really a spider web of interactions, and that plays out as we think about some of these more complicated diseases. Let's start with the simpler case, though, where we have complement dysfunction as one of the major contributors. And there are several diseases where complement dysregulation is implicated as a main driver. That's shown a little bit here on this right graphic, where you can see that there are multiple organs, kidney, red blood cells, the eye, the brain, that have been associated with diseases in these organs have been associated with abnormalities in complement regulation. And I'll just take one example. So in the bottom left, we're looking at red blood cells. The disease is paroxysmal nocturnal hemoglobinuria, or PNH. And it's characterized by an increased risk of complement-mediated lysis of red blood cells. And that is associated with deficiency in some of the regulatory proteins that operate in the complement cascade. And so this is just, one example of a place where complement deficiencies are sort of directly trackable to diseases. Okay, so what are we really talking about with respect to complement? So in this figure, what we're looking at is the possibility that complement can be activated via multiple routes. And so complement can be considered in terms of a classical pathway, lectin pathway, and an alternative pathway, each of which are sort of stimulated or activated or kicked off in a slightly different manner. They converge in this sort of middle bottom box down here at convertases, which are sort of responsible for the terminal pieces of the pathway, which then drive inflammation, lysis. So we were talking in that previous slide about how in PNH you have hemolysis. Well, this is sort of one of those terminal pathway effector mechanisms, as well as opsonization. I'm going to spend just a minute talking about the alternative pathway on the far right. This pathway is interesting because it's characterized by a couple of features, namely tick over and amplification. So tick over is sort of, you can think of that as sort of this low-level activation event that occurs and is then, regulated by all the regulatory factors to keep your body from going into an inflammatory state. And it basically provides like a low-level hum, like the motor is running and ready, so that if you end up having an infection or something else that kicks off activation, say via the classical pathway, the alternative pathway is ready to go. The other aspect is that the alternative pathway has an amplification loop indicated here on the sort of middle right, and that allows you to get more bang for your buck. So you have sort of maybe an initial stimulus, and then the complement pathway can really ramp up, and that ramp up has been estimated that to account for up to roughly 80% of the total complement pathway activity. So even if you are stimulated through the classical pathway, the robust complement response that you get out the other end is largely due to the alternative pathway. Okay, so why do we think that it's going to be therapeutic value associated with the complement pathway? Well, this has already been shown with several treatments that have been approved. So starting with eculizumab, which targets C5, that's one of the complement proteins that is cleaved by a convertase that I mentioned in the previous slide. That was approved by the FDA for the treatment of PNH in 2007. So PNH, the disorder where you have lysis of red blood cells. And then subsequently, eculizumab has been approved for other indications. And then, and that is I think a continuing story that we'll watch evolve over time. And there are other approved as well, ravulizumab also targeting C5, and pegcetacoplan, which is a recent approval in 2021. It actually targets C3. So if we're looking at this figure on the bottom right, C5 is sort of the targeted by the C5 convertase. C3 is a little bit upstream and targeted by the C3 convertases, but these all have the potential to, and clearly have been shown to have benefit for patients. These three, given the successes of these three, it's not surprising that there are many other compounds in development. Not all of them are shown here, but this is sort of a sampling where we can look at, again, the complement pathway, and you can see down here is a terminal pathway where eculizumab is operating, the alternative pathway in purple, and then the classical and lectin pathways in blue and yellow leading to green. So multiple other compounds in development. Why is this sort of, so given the intense interest, why is this something that is also challenging to develop? So complement has, multiple proteins that are involved. Some of these proteins are present in relatively great abundance, and so there's the question about how much do I need to knock it down. Some of them are characterized by relatively rapid turnover, so you might have questions about how frequently do we have to dose in order to effectively reduce the levels of this particular protein. We've talked about the amplification loop and the regulatory mechanisms, and there are many feedbacks going on. So tracking all of that and sort of having a clear understanding of what you might expect to see at the back end is, has its challenges. And then finally, this is part of the innate immune response. There is sort of this, potential risk that if you knock complement down very robustly, that you might increase the susceptibility to infection risk in patients. So before we go on to the QSP model, what I do want to also sort of just pay a moment of attention to is that in addition to these relatively rare diseases where complement is sort of the primary lesion, there are also sort of an increasing appreciation that complement may be playing a significant role in the pathophysiology of other diseases. And that includes non-alcoholic fatty liver disease, cancer, the hepatocellular carcinoma, as well as autoimmune diseases. And sort of to illustrate this, we have a graphic of non-alcoholic fatty liver disease. There's histological evidence of complement deposition in the liver. We know that this is a metabolic disease, but adipose tissue turns out to be a major source of complement proteins. And as well, we know that NAFLD is in part characterized by inflammation. So complement, in addition to the lysis pathway that we're sort of focusing on for PNH, there's also the inflammatory pathway mediated by C3 and C5a. And so that is also a way in which it can directly impact the liver. There's also some evidence that there may be some association with insulin resistance. And so lots of opportunity to maybe expand beyond these, relatively rare conditions where complement dysregulation is highly implicated into other areas where complement may be a major player, but not necessarily the causative agent or the only player. Okay, so why are we interested in QSP to address complement development? Well, many of you will already be familiar that QSP is a mechanistic representation of the patient physiology or pathophysiology. It is the interaction of the drug exposure and drug mechanism with that pathophysiology, as well as accounting for interpatient variability in those mechanisms in order to predict efficacy. And sort of some of the challenges that I had talked about in terms of what targeting the complement cascade, some of the things that we would think about in terms of feedback loops and the amplification loop and how to manage that. That is very amenable to mathematical modeling to sort of put, the structure of math in place to really constrain what you're looking at and give it that rigor to allow you to interrogate a system in a way that is systematic and is, robustly includes the available data that we have to characterize the system. The other aspect that you can think about is that in these QSP models that you are leveraging the available data. And so this can be considered sort of a repository for what are all the things you know, and then where are places where we don't know that much? And are those places important in our predicted efficacy, and therefore places where it would be fruitful to collect additional data? Okay, so I did mention that there's a lot of data out there, and just to give an example of this, this table is from several decades back now, but this is an in vivo tracer study to look at metabolism of C3 in people. So that type of, goes back to that whole comment about, using the QSP models as a repository for collecting all sorts of data that you have, putting it together into an integrated platform, being able to really leverage the scientific community's, global knowledge. I will also acknowledge that we are not the first people to investigate complement via QSP modeling. There are several groups that have worked on this in other contexts. Sometimes, there's a different focus, acute versus chronic, or which particular pathways are being represented or to what degree. But I think what, the point I'll make is that this is an area that is A, amenable to modeling, and B, an area where the benefits of modeling, I think, are very, very much appreciated. Okay, so let's shift gears and talk about what we are doing. And don't worry so much about trying to read the graphic on the right, since I will show that in the next slide in larger scale. But the idea is that we are representing a base model that is focused on the alternative pathway and the terminal pathway. You'll recall that I had mentioned that the estimates were that the alternative pathway accounts for roughly 80% of the total complement activity. And so this is an area where, our initial foray, it makes sense to sort of focus here. We do have a limited representation of say, a classical pathway to initiate and look at disease conditions. But the focus is really alternative and terminal. And then what we're doing is we're developing alternate simulated patients that reflect inter-individual variability. We're comparing that against the available data in normal healthy volunteers, and then also looking at that same sort of exercise, but looking in disease patients. We are also including 3 exemplar compounds, so looking at eculizumab, targeting C5, pegcetacoplan, targeting C3, and iptacopan, targeting factor B. And these allow us to sort of interrogate the model in evaluating it against published data in order to sort of build confidence that it is representing what we think it's representing, and that it is sort of behaving in a way that is cohesive with the available data. And then the idea is that, so you can, even with the base model, represent the disease state by looking at those complement proteins and regulatory proteins that have been measured in disease patients. But we are also looking, and that would be a customized model, right? Focused on a disease representation rather than normal healthy. But we are also looking to be able to expand this model to be able to look at target tissues. And so, for example, if we look at, say, the focus was PNH, and for understanding the influence of hitting particular complement proteins in PNH, you might want to represent red blood cells more specifically. So represent that red blood cell membrane, the interaction of complement proteins with that membrane, what changing those would, what impact that would have on hemolysis, and then subsequently, on things like number of patient transfusions. So things that will really be helpful in answering questions about PNH. And we could do that for multiple systems, kidney, eye as well. And then I had also mentioned that if you knock down the complement system very robustly, you do have the potential to compromise your susceptibility or your to infection. And so there's also the possibility to expand into the area of safety. So as promised, this is our current QSP model focused on the alternative and terminal pathways, but in a sort of larger scale. I'm not going to go through all of the details here, but I will say that we have, the representation of C3, C3 convertase, C5 convertase, the cleavage of C5, and the terminal pathway that can be linked to functional or clinical endpoints. Okay, so what do we do with this model? So I had mentioned that we're integrating the available data. In addition to just sort of an integration, there's also the piece where you can test your model against in vitro data to ensure that it's behaving in the way that you anticipate it should be behaving. And so, for example, you could take in vitro data as shown in the bottom left here, where essentially they're looking at the decay in spontaneous decay in C3 hemolytic activity shown here at the top. And then if you add in the activating factors, factor B and factor D, you see a much more rapid decay. And that is mitigated if you add in the regulatory proteins, factor I and factor H. And so we can set up, the same sort of experimental conditions in the model and be able to reproduce those behaviors, which then gives you the confidence that you are, in fact, putting things together in a way that is consistent with in vitro data as well. And then beyond that, we want to actually be able to compare with in vivo data. So what we're looking at is a handful of analytes here. This is not all of the analytes that we do comparisons for, but for the purposes of space, I'm only showing 6 here. So basically what we've done is a pretty robust evaluation of the publicly available literature and pulled out data for, say, on the far left, factor B levels in normal healthy volunteers. And so we can plot those data, and then what we're looking at across the board then is for each of those analytes, what is the published data telling us about the median, the range, depending on the particulars, and then how is our initial simulated individual doing relative to those range? And so that's the black triangle. Is the same simulated individual across these different analytes. And so, the idea is that you want to be able to sort of feel that your individual is in line with the measured complement levels as seen in the published literature as a starting place, right? So that is one piece of the puzzle. I guess in addition to that, that, hey, we want to know that not only is sort of the steady state baseline levels, but what happens when some of those levels go down or or altered, as you might expect in, say, a genetic condition. And so we have identified multiple publications in which people have reported individuals, cases where they have factor H deficiency. And then they've looked at other complement proteins. And so in the figure to the right, what we're looking at are sort of a summary of some of these literature. On the sort of the right-hand group are the controls reported in that literature. So these are typically family members that do not have the particular factor H mutation. And then on the left are the cases where you have some level, and this varies by case because it varies by mutation, of factor H deficiency. And what we can then do is in our initial simulated individual, we can drop the factor H levels and see, do we see a corresponding decrease in C3 as was seen in the cases? And in fact, we do, and this gives us increased confidence that the model is appropriately representing the relationship between factor H and C3. So the other thing that we do is that we try to represent individual variability. And so more specifically, we take our complement model and we identify some of the parameters where there is reported variability or where we anticipate a certain amount of variability. We impose that, and then we generate alternate simulated individuals. And then we can evaluate those individuals against the clinical data. And then in the instance where we're going through the same exercise but creating diseased individuals, we can also evaluate relative to therapeutic response for the treatments that we're adding in. So what does this look like? Here's an example here where showing that for C3 on the left, C3A in the middle, and soluble C5b through 9 on the right, that the literature data shows us a range left in each panel, and then the simulated individuals shown in the red give us a distribution that we can look at across and see that we're covering sort of the range that is reported in the literature. And then finally, what we would do then, this was all normal healthy comparisons, but what we then do is we take our, we start thinking about diseases. How do we represent diseases, even if we're just focusing initially on that circulating compartment? So I'm going to use published data as my example of this because we can't disclose that for the pharma partner. But say, for example, that you were interested in C3 glomerulopathy. And so we would identify the data in the literature that would characterize these analytes, of complement analytes in patients with this disease. And so what we're looking at on the right is various analytes, so C3 in the top left, C3A, BB, C5, and it's comparing normal healthy with patients with C3 glomerulopathy. And so we can generate patients that align with these data. If you have proprietary data on C3G, we could also generate patients that align with proprietary data. Again, that would be in a custom model, not in the base model. And then we can also look at the responses to various treatments as part of the qualification of those patients. Completing that cycle, then we would move forward into evaluation of novel treatments or looking at, exposure and mechanism of action and, different dosing protocols to see what is the impact of targeting a particular protein and reducing it to a particular level with these kinetics in order to sort of estimate what the predicted efficacy would be. So with that, I'd like to thank our pharma partner for making this possible, but also the complement development team who have invested an enormous amount of effort and work in putting all this together, as well as Jonathan Chauvin from our Lisl division, who has helped us with the representation of PK for some of our treatments of interest. And with that, I know we are close to time, but I'll turn it back to you, Andy.
Lisl, beautiful presentation and fantastic timing. Thank you, Lisl, and thank you all for joining us. Reminder at the end of the discovery and preclinical track today, there will be a group Q&A session, including perspectives from all of the track speakers to address any remaining questions you might have. Please be sure to stop by. This concludes Lisl's session. Please leave this session, head to the lobby for the next presentation. Thank you for staying with us. Thank you, Lisl.
Thank you. Bye-bye.